go to joinpd.com. I'll get the code here.
Looks like there's a question. Sarah's got her hand raised.
What's your question, Sarah?
Yeah, I think you might have already answered this, but I'm still having trouble looking for it. Where exactly is the research and paper instructions on Canvas?
Presentation instructions. If you go to modules, it's in the last module. At the bottom, it's called something like ASRI assignments. And it says something about PowerPoint presentation.
Right there, where you are on your screen, you should have what you wanted. Do you see it? On your screen, you should see what you wanted, what you were asking me about. Do you see it?
ASR assigned to submit here on Canvas. That? Oh,
you don't need to submit any right now. You just can, those are just instructions for how to be working on it.
Oh, okay, I see it. Thank you.
Whitney, did you find that?
Yeah.
Okay.
I think.
Or it's right there.
I'm just not sure if there's an easy way to get it on my phone.
It is. It's a website that pops up on your screen. Right there. app.peardeck.com/dash.
Okay.
Okay, you guys, go ahead and get on. I know if you're having trouble answering questions, I can't give you points. I mean, that's the bottom line. So I do go through that meticulous process of checking. And if you answered most, I give you credit. If you answered 1 out of 4, I don't. So it's just a matter of most. And Whitney, it'll tell you how many students have joined. And John, you asked about Labster. Again, there are 4 other links on in the schedule that I showed you, you know, like the NODAC animations or the HHMI interactives or biointeractives, or there was the, you know, kind of the very first one from University of Utah, how to do PCR or microarrays. So please pick something and you can submit that because we knew that Labster will challenge your Wi-Fi and computers.
Maria, did you have a hand up?
I was asking for I was going to ask for the code, but I see it now in the screen.
Yeah,
Sorry.
Yes, right up here. Thank you. Okay, we have 47 students joined. I think that's probably most of you. So I'm going to go ahead and
that code will remain in the corner, so you can still keep logging in.
Yes. Okay, so here's your first Pear Deck question. Have you given a presentation before?
Like since coming to college or like in general?
In general, if you feel like you gave a presentation in high school that was, you know, well done, that lasted at least 8 or 10 minutes, then I think I would count that. Okay. I'm going to show the responses here. We have a lot of experts, right? I'm glad to see that you guys have done, that most of you have done at least a couple of presentations. But even for those of you who said no, never, you know, this is a great way to do it because this is kind of a low stakes environment. We're not grading you. You'll get lots of feedback. And so I think that you'll have a really good experience. In your opinion, what is the hardest thing about presentations? think there's a couple of students that just come in from the waiting room. If you'll go to joinpd.com and enter the code that's in the upper right of the screen here, JSKAD. You can also find that code in the chat box. I am going to go ahead and show the responses and see what some of you are saying. Nerves and the initial time commitment, remembering words that are hard to pronounce. That's definitely something you have to practice. Remembering stuff, handling large audiences, speaking in front of people, keeping your pace, you know, fear of speaking in front of large groups, standing in front of people, being prepared to answer any questions. This was always hard for me because I felt like, what if I just get that curveball question and I don't know the answer and I look silly? Having something the judge pulls apart, wanting to have confidence. Yeah, these are all, I think that you can see that nobody says that there's nothing hard about it, right? So I'm going to jump in now with just a lot of advice and tips for putting together your presentation. You'll want to start with some kind of title slide. I have a picture of the one that I used right here. Make sure that it's a title that explains your topic. If you've got something catchy, that's great, as long as it's still professional. We usually recommend that you don't read your title out loud, particularly if it's the title of a paper or a research title and it's 15 words long and it's a study of the ontogeny of antimicrobial peptides in the southern leopard frog, Rhanus phoenocephala, up to 12 weeks. Like, you don't need to read the title. You should paraphrase it. You should say, I have been studying development of antimicrobial peptides in leopard frogs, and I'm going to tell you today about my findings. We also want you to practice introducing yourself. You should say at the beginning something like, hi, I'm Sarah Smith, and today I'm going to tell you about my studies involving antimicrobial peptides in leopard frogs. But you should also practice if somebody introduces you. I've seen students before, they've practiced introducing themselves, but when somebody else says, and now Joseph Jones is going to tell us about antimicrobial peptides, then Joseph Jones stands up and says, hi, I'm Joseph Jones, and I'm going to tell you about antimicrobial peptides. And they just kind of repeat their own introduction because that's how they've practiced it. So practice introducing yourself, but also practice if someone else introduces you saying, thank you, Dr. Holden. Hi, everyone. And then going from there. Also, make sure you're using appropriate background images and logos, be professional, consider your audience. This means that if you're giving a presentation to a group that knows a lot about your topic already, for example, at a professional research conference, you might not need to spend as much time introducing, for example, the terms that are in your title. Whereas if you are teaching a group that isn't going to know much about your topic to begin with, you might have to explain, antimicrobial peptides. Those are just the peptides in the skin that are antimicrobial. Now, a question for you guys. When you are doing your presentations on next Friday, you will have an order to your research presentation. So whether you are presenting your own data or you are presenting a paper that you picked on the first day, you'll need these sections in your presentation. And what is the correct order of them? Okay, 5-second warning. I'm going to lock the screen. Okay. So we see an overwhelming choice for selection D, and that is the correct one. You start with your background first, sometimes called an introduction, actually probably called introduction more. So we usually call it introduction, but it contains all of the background. There's usually a hypothesis at the end of that, a brief discussion of methods so that they understand how you got the results. We don't want to spend too much time on the methods though. And then finally, what the conclusion is from those results. Okay. So with that introduction, this is where you present your purpose. So you start off with that purpose or that problem statement. Tell your audience why you're doing this. I am studying antimicrobial peptides in southern leopard frogs because I want to know how or even if these peptides help protect this species from the fungus Batrachochytrium dendrobatidis. Then you go into your background information. It can be one slide, it can be 2 or 3 slides, as long as you don't have too many words on those slides. You don't want to spend your entire time on your introduction. This is where I would introduce the southern leopard frog, the fungus, the fungal pathogen Batrachochytrium dendrobatidis, the symptoms of that fungus, how the fungus has spread around the world and is causing major amphibian declines, how it seems that there are some frogs, including the southern leopard frog, that are a bit more resistant. And then I'm going to jump into my hypothesis or goal or objectives, depending on, some of you won't have a hypothesis. Some of you will have a goal to develop a new prosthetic design or to develop a wearable device, or, objective. You might have sort of 2 or 3 different objectives that you're trying to accomplish within a project, or the paper might have a few objectives. But these are all, these are all going to come right after you give that background information. This is where I would say my hypothesis is that the southern leopard frog will have a large amount of diverse antimicrobial peptides in its skin that are capable of fighting off this fungal pathogen, both in vitro in a test tube when they are purified, and also in vivo, so that the frogs that have those antimicrobial peptides will be able to fight off this fungal pathogen. And those that have their antimicrobial peptides removed before being challenged with the fungus will have greater infection. So that's where you would discuss that hypothesis. This point right here, use parallel sentence construction for readability. That just means a couple of things. One is to make sure you're using the same tense. If you've got 4 bullet points on a slide, you shouldn't have 2 full sentences and 2 fragments. Generally, we prefer fragments that are short and readable. You also don't want to be skipping around between tense. So you don't want a couple of bullet points that are in present tense and a couple of bullet points that are in future tense. Keep things parallel, keep things consistent. Also use consistent punctuation. If you are using a full sentence, it does need a period behind it. If you are asking a question, like if a bullet point says, how could this be? It needs a question mark at the end because that's a question. Then after you have introduced your topic and done your hypothesis or your objectives or goal, you want to introduce the methods. In general, I recommend limiting this to 1 slide if possible. Sometimes there's more than 1 slide that makes sense. For example, if you extracted DNA and did PCR, you could potentially have a slide for the DNA extraction and a slide for the PCR if you have a clear break like that. But we don't want the methods to be overly detailed, and please, please, please don't put your entire method section on a slide. I've seen many, many students do that. They take the methods and they've got 14 bullet points on a slide, and they're using size 10 font, and it's no good. Nobody can read that. Your audience is squinting at the screen trying to read it. If you can use pictures, that's really nice. So for example, up here I have a picture. This is from my undergraduate thesis work at the University of Arkansas. I was using photoisomerization. I've got this UV light right here that's in this chamber, and there is oil that is being exposed to it. And this is one of the steps where I was trying to make a compound called conjugated linoleic acid. So I've got a catalyst in there, and the UV light is helping to make that conjugated linoleic acid. Right here is a slide that I used through my graduate work, where I took a ton of different methods. And I actually, I took this picture, and all of these things I just made in PowerPoint, except for that one right there is a picture. And this one right here was made by another person in our lab. But again, they did make it using PowerPoint. And so I've just got, I could say, how do I collect the antimicrobial peptides and analyze them? I take the frog, I inject it with norepinephrine at 40 grams, or 40 micromolar per gram body weight. That's a pharmacological dose. This is much more norepinephrine than they would create physiologically, and that creates all of those adrenergic receptors to fire, and all of the antimicrobial peptides come pouring out of those glands. So I put the frog in a collection buffer for 15 minutes. This has salts to stabilize the antimicrobial peptides. I then inactivate the proteases with 1% trifluoroacetic acid, and I can go through this. And it's with pictures. And so they understand that, I'm blasting a laser at these peptides to get a plume of ions so that I can determine the peptide sequences. And this is so much nicer if you have the ability to do this. Now, this is maybe something that you can't do very well yourself, especially if you're not doing your own methods, like you're not working with your own data. You might not really understand the methods well enough to be making images for them, and that's okay. But you should still try to have just bullet points with fragments. Frog was injected with norepinephrine to release peptides. Peptides were enriched with C18 set packs. Multi-TOF mass spectrometry was used to identify the peptides present, and growth inhibition assays were used to determine if the peptides were effective at killing the fungus. Just like that, very brief. Now let's talk about the results section that will follow. When you are presenting your results, you want to make sure that your audience can understand them. And an important part of that is making sure that graphs and tables and diagrams are understandable. So I want to see if you guys know the pieces of a graph. So you can drag these icons here to label the x-axis, y-axis, the legend, and where the graph title should be. And while you're doing that, take a look at this graph and notice all of the things that are wrong with it, because there's a lot wrong with this graph. I can see all of your icons moving around, and you're doing a good job. I'm going to give like a 10-second warning to finalize your answers. Okay, and then I'm not going to lock the screen on this one. I'll just show the responses. We've got the red stars down here. That's the x-axis. That horizontal axis is the independent variable. This is the thing that you are changing. So the different concentrations that you're using, or the different treatments that you are using with your groups. We've got the orange heart on the y-axis. That's the dependent variable. That's the thing that you're measuring. So if you are using these various concentrations this is whatever you're measuring, whether it's like plant growth or bacterial growth, or some kind of metabolic pathway that's being activated that you're measuring. We've got our legend over here. This is what tells us what the red squares are and what the blue diamonds are. And there are actually a few, like I see one green triangle here, and that is where you might often find a graph title in a paper, right below the graph. You would see a figure legend with the title down here. But in a presentation, we usually do see the title up above the graph. So with graphs, you need to take the time to explain the graph, particularly when it is a little bit more, when it's a little less self-explanatory. I always start with the axes, and it's important those have to be labeled. This over here, I give a big sad face. There's nothing labeled. There's no way to understand what any of this data means. Here, we see that the x-axis is actually measuring the length of the conductive paper. And here we're measuring the resistance on the y-axis. I've got my units. So we've got length in meters, resistance in kiloohms. And then my legend is filled out, and I know that this is 2 centimeter wide conductive paper, and this one's 2 centimeter wide conductive paper, and this is 1 centimeter wide conductive paper. And I can see, so I've got everything labeled. And if I am presenting this to a group, I start out with the title. I say, here is my data showing the resistivity of conductive paper. You can see on the x-axis here, we have increasing length of conductive paper. This data set right here is for 2 centimeters wide. This one here is for 1 centimeter wide, and I'm measuring the resistance in kiloohms. So there, I've told them all the pieces. Now I can say, and you can see from this graph that the 2 centimeter wide is less resistant at all length, less resistive at all lengths measured than the 1 centimeter wide paper. And even that the 1 centimeter wide paper gains resistivity faster as the length increases, which you can see from that steeper slope in this series versus this series. So label those axes, explain the important elements, and it's okay to pause for a couple of seconds while they digest this information here. Then after you have presented your results, you want to explain outcomes. This is your discussion section. This is where you say, was your hypothesis supported or not? Or did you accomplish your goal or your objectives? Also, what else did you learn that went beyond your hypothesis? So for example, perhaps your hypothesis was that if I go back a slide, perhaps your hypothesis was that the 1 centimeter wide paper would have greater resistivity, but perhaps the fact that that slope was steeper, perhaps that was something that you had not expected. That was a surprise, and it's worth noting in your discussion. This is also where you can say what still needs to be done, your future directions. I plan on extending this both to even wider paper, 3 centimeters and 4 centimeters, and I'm also going to be testing some different materials to see if the same tenets hold true, if the thinner paper really does continue to gain more resistivity even when it's made out of a different material. You can also mention weaknesses in the work, sources of error and unexpected difficulties. Sometimes sometimes I see this in certain fields and not other fields. When in doubt, I would say ask your advisor if they want you to mention sort of unexpected difficulties. Dr. Krakowiak made a good point yesterday. For sources of error, don't talk about things that you can help. You don't say something like, I think that there was some error introduced when some of the paper that I used got wet. Well, don't do that. Redo it with paper that didn't get wet. So other sources of error might be things like, the machine itself can only measure up to, 0.1 kiloohm. And so some of the differences, there may have been minor distances that couldn't be read by that machine, and that's why you're going to be spending next semester going to UALR to use a more sensitive machine that they have there to continue to collect data to see if it supports or refutes your hypothesis. There might also be sources of error with, for example, when you handle the paper, you have to handle the paper to put it into the machine. And initially you found that when you were handling it and there was residue from your fingers, that might have been contributing to some of the differences in numbers. And so then you went to using latex gloves, but then there might have been some latex being transferred as well. So then you were using tweezers, but when you were using tweezers, that made it more difficult to tear the paper. And so this is something that you're still working through, that it might be appropriate to say something like that as well. I see the chat box is going crazy. I assume it's being monitored. Here's another Pear Deck question. Walls of text should be avoided. What do you guys think? I'm gonna give you 5 more seconds to get your answer in. Okay, so true, yes, walls of text should be avoided. And some say, I have no idea what this is. I understand, so I'm going to show you. This is a wall of text. Do not do this. Do not have something like this on your slide. That's just a big old paragraph. Your audience wants to read it, and your audience is trying to read it, and they're trying to listen to you at the same time. And they're not going to be able to listen to you effectively enough to comprehend you, and they're not going to be able to read this with you talking enough to comprehend it. And so they're not actually going to get very much off of this slide. We want bullet points with fragments instead of tons and tons of stuff to read on a slide. We also want you to use images in an effective way. So I sometimes see slides that it's just slide after slide after slide of text bullet point, and there's not a single picture. I've seen students give a presentation where they're talking about a particular organism, maybe a microorganism, maybe a vertebrate, and they don't even have a picture of that organism. Think about on each slide what you can do. On a method slide, if you're using electrophoresis, have a picture of a gel on the method slide, just so people can see what your methods look like. This makes it really good for visual learners. It makes the slides look very nice and professional. I do want to say use care regarding copyright. You can't just be like, well, I'm studying, this particular kind of food additive, so I'm going to go and get a picture of a product from a Google image search with that food additive in it, because it might be a copyright image. And so I often will get images, either, ideally you would take them yourself if you could. Like I took a picture of my own frog or a picture of my own micro-BCA assay. But if you can't do that, you can go to Wikimedia Commons and look there in the credit, and always give credits to the original author of that picture who's sharing it. You can also use something called freepik.com. I got this from freepik.com. It's free with attribution. And so I just have kind of covered up, I think, by the Pear Deck there. So source freepik.com, and there's a number and then the person who made it. So I can use that to make my slide a little bit more enjoyable. When you have a very complex image, Dr. Krakowiak referred to this one a day or two ago, take time to explain the complex images. So for example, I've got a circle here that contains all of human knowledge. And this is how much you're learning in elementary school, in high school. When you gain a bachelor's degree, you've got like probably a nice liberal arts kind of education, but a focus in one area, that's your major. A master's degree deepens that focus. Reading research papers takes you to the edge of human knowledge. When you're at the boundary, everything there is to know about this little slice of the world's knowledge that you're interested in, you push at the boundary, and you learn a little bit more. And that's your PhD. And the world looks different to you now. Because you've spent 5 years working on a PhD and you've added all of this knowledge. But this picture sort of amusingly tells you, don't forget the bigger picture. You've added this wee little bump right here. But I say keep pushing, because you are widening the circle. You're making that circle bigger and bigger for all of our knowledge to fit in. So that's just an example of explaining a complex image. With transitions, sometimes people will use a starting outline at the beginning. This typically isn't necessary unless you have a very long presentation. If you're giving an hour-long presentation, it might be nice to have an outline at the beginning that says, I'm going to have 3 sections where I discuss these 3 things. If you're going to have an unusual structure, you might also mention that at the beginning and say, I'm actually going to start with my conclusion, and then your reason for that, although that's typically not something that you would see. We also don't want you to read your slides. We want you to explain, put things in your own words. Anybody can read your slides. So if you are up there just reading from your slides, you're not helping your audience understand, and we want to hear what you have to say about things. You can enter a number here. How many minutes should you spend on each slide in a presentation approximately? What's the general advice? Gonna lock the screen. This is in minutes, not seconds. I see a few of you that maybe have, and this is on each slide in a presentation, not on the entire presentation. Okay, I'm going to show the responses. So looks like most people are saying between 1 to 2 minutes per slide, and I would say that that's fair. I think that the typical advice is usually 1 minute per slide. Of course, if it's like a title slide, I don't spend that much usually. If it's the most important slide with the most important data of your entire presentation, I think 2 minutes would be okay for that. We also want to make sure that you place emphasis correctly. There are various ways that you can emphasize to your audience what is important. How much time you spend gives weight. You shouldn't be spending 5 minutes on a method slide because the methods are not the important part. The data is the important part. Words also give weight. So if you say things like, interestingly, we weren't expecting this. If you're going to take one thing away from this presentation, this was the most critical part of this project. You say those kinds of things, and your audience knows to pay attention to this sentence, that they're getting a really important nugget of information. Also, repeating the most important points is a good idea. So this can be done visually in that you could have a bullet point about something, but then you can also show a diagram emphasizing that thing, so you have a visual backup to the point. And then saying things out loud. If it's fine to say during your results and during your discussion whether or not your hypothesis is being supported, because that's something that you really want to get across to your audience, what your general finding is. Another Pear Deck question for you. An important key to success for a presentation is to do what beforehand? If you guys don't know what a power pose is, there was some research a few years ago that I think is still somewhat tentative, that you could go into a private space, into like a closet or an office or a bathroom stall, and assume this like really powerful like warrior pose for a minute, and it would psych you up. So that's what the warrior or what the power pose is. Okay, I see a couple of students have been joining us. We're nearly done, but if you want to hop on Pear Deck, you certainly can. The code's in the upper right. I'm going to lock those screens now and show responses. Most people say practice. Yes, I do understand. I have prayed before big presentations before, particularly a qualifying exam, lots of prayers before a qualifying exam, which determines whether or not you can continue with your PhD work. Drinking enough caffeine, also quite important, especially if you are used to being regularly caffeinated, but I think most of you know that practice is key. So practice makes perfect. Practice more than once. Use a timer. Make sure you know if you are the type that speaks really, really fast when you get nervous, or if you speak more slowly when you get nervous. Practice the transitions between slides, and practice with another person who can ask you questions, who can interrupt you, especially if I had a group of students a couple of years ago that they memorized their presentation, and they said their presentation exactly the same way every single time. But then they were more likely to get flustered if they were interrupted. And so practice with someone who can interrupt you, who can introduce you, who can ask you questions. And the more people you practice with, the better. If you guys want to practice with each other before Friday, that might be a good idea. If you know each other and you're willing to maybe share a phone number and practice with each other via FaceTime, that could be a great way to practice for your presentations at the end of this week. Now I have a question for you. Do you have any advice about presentations to share with your fellow students? So we're going to be we've got to move on to entrepreneurship here in a quick sec. So I'm going to give you guys about 30 seconds just to type advice for giving a good presentation that you have. see a really good one that several people are talking about. Couple of really good ones. Yeah, let me share and show you guys. You can keep working. Give yourself time, get a good night's sleep. That's a really important one. Get enough sleep beforehand. Be confident, know you have the information. Breathe, take deep breaths. Pretend you're confident even if you're not. That's such a big one. I've seen so many presentations where it's clear that people are nervous, and I understand because I've also been that person who has appeared very nervous when I'm speaking. This is your project, and you've done a great job, and you've worked hard. So some people use note cards to speak off of. Take some deep breaths. Deep breath, drink plenty of water. Focus on your presentation, not your nerves. That's a good one. Get to know your topic really, really well. Pronounce things. Enunciate, carry your voice. Another one for breathe. Put the most important point on top of the slide and enlarge. Yes, we want the fonts to be large enough that people can see them. That's another way to give weight to something, to indicate that it's important if it's in a larger font. Look around the room once in a while. I always try to pick out those friendly faces in the audience, somebody who's nodding and looks kind of friendly, and I make eye contact with them when I'm looking at my audience. Practice, 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 more practice. There's lots of really good ones. None, because I'm bad at presenting. I hope you have learned some tips now. Everyone's been nervous before. That's true. An excited presenter helps the audience stay engaged. These are really, really good. I'm going to go ahead. It's only 0.005% of your life. Don't sweat it. That's another good one. I had a cat in grad school, and I would tell her before every test and before every presentation, I would say, like before I left the house, I would say, when I come home, it's going to be in the past. And then I would leave, I would do it, and I would come home and be happy with my kitty cat. Okay. So I want to tell you guys about something really, 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 really cool. This is called slidesgo.com. There are various websites, and this is one of them, where you can get free slide themes online that you can download and that you can use for free as long as you attribute them on your in your acknowledgement slides. And so I really encourage you guys to go to slidesgo.com. There are medical templates, there are engineering templates, there are computer templates. We have used SlidesGo templates for various presentations in this during this ASRI, and you can get some really cool ones. During the next break, I will open up SlidesGo and just show you guys more about that. Because I think it's a really great way to get a very professional looking presentation. And so for homework, what we want you guys to do is to actually pick a theme from SlidesGo or from another free template website and start building your presentation for next Friday. Tomorrow in breakout rooms, we're going to let you guys show off a couple of the slides that you've been working on. So maybe the slides for the introduction section, that'd be easy enough. And then each room is going to vote on the coolest, most professional looking slides for earning a point towards door prizes. So again, during the next break, I'll go to slidesgo.com. I'll show you how to download them. And this is a way that you can earn some points and get some cool free resources. And thank you very much. Do you guys have any questions for me?